Human blood types. Blood transfusions, the administration of blood directly into the bloodstream of another person, may seem like a miracle of modern medicine, but the concept is not new. For over a century, physicians have tried to counteract severe blood loss by transfusing blood from one living person into another. Sometimes these early attempts were successful. More often, they were not, resulting in severe illness or even death for the recipient. Why did the first attempts at transfusing blood save some lives but not others? Today, we know the success of blood transfusions depends largely on blood type, based primarily on the ABO blood group system. If you ever donate or receive blood, you will undergo testing to determine your blood type. This is necessary because if you receive blood from someone who does not belong to a compatible blood type, you could suffer a severe reaction. To understand the concept of blood typing, you must first be familiar with antigens and antibodies. Our cells have certain surface proteins that the immune system can recognize and identify as self, in other words, belonging to us. These are like passwords that cause our immune system to ignore our own cells. Foreign cells carry different surface proteins, which the immune system recognizes as non-self. An antigen is not no, an antigen is a non-self cell protein that stimulates the immune system of an organism to defend the organism. As part of this defense, the immune system produces an opposing protein called an antibody. Produced by lymphocytes, antibodies belong to the class of plasma proteins called gamma globulins, mentioned earlier. Antibodies mount a counterattack on antigens as they recognize as non-self. There are many antibodies, each one specialized to attack one particular antigen. This response has been compared to a lock and key. Only a specific antibody key can fit a specific antigen lock. Antibodies float freely in the blood and lymph until they encounter an invader with the matching antigen. They bind to the antigen molecule to form an antigen-antibody complex that marks the foreigner for destruction. The formation of an antigen-antibody complex often causes the foreign cells to clump together, effectively inactivating them.